Hello, I'm Ina, a biomathematician currently finishing a PhD in applied mathematics in Barcelona. Here, I want to share with you my idea of constructing dynamic microbial association networks in the ocean. And you can also see what a mathematician is doing at the Marine Science Institute. Microbes are everywhere, not only in your gut helping you to digest your food, they are also in the ocean. They are key players in major nutrient cycles. Did you know that on average, every second breath you take contains the oxygen produced by the microbes in the ocean? These microbes are important, but we don't know enough yet, and we would like to know who is interacting with whom. They are so tiny, you can't observe all interactions with the eye or microscopes. Luckily, the technology, more precisely high-throughput omics technologies, advanced tremendously over the past years. We are now able to determine who are these microbes, and even further, we can count them. Using these counts, we are able to infer microbial interaction hypotheses. My collaborators are super interested in the microbes, and they should, given their importance. And I am fascinated by networks. When I think of microbes, I think of a network similar to the social network. Each node is a microbe, and if two microbes interact with each other, for example, they help each other in a symbiotic way, one is eating another, or host-parasite relationships, they are connected by an edge. Having such a microbial network will help us to study the microbial ecosystem. For example, we can infer important nodes. Importance is relative and always depends on your definition. There's no ideal way how to detect keystone species in an ecosystem through a network. One way is to look for hubs, that is, highly connected nodes. Here, the node with the crown seems very important, and the node below unimportant. This node has only two edges, but it functions as a bridge between two modules. So for network architecture, this node is very important, and it is up to the biological interpretation to find out how such network importance translates to the ecosystem. Speaking of modules, a module is a group of highly interconnected nodes. And then we would investigate why are these microbes more connected? Are they involved in the same nutrient cycle? Do they share the same environmental niche? This network here is just an example. My network actually looked like this. I had a time series of microbial abundance data for 10 years from the Mediterranean Sea. From the monthly sampling, we obtained 18S and 16S rRNA data, which we used to construct the network. It didn't really look as pretty as you can see here in the beginning. In fact, we first obtained a hairball, a dense network with way too many edges. We applied a filter strategy, including the removal of environmentally driven edges, to get a network that is less dense. What I didn't like was the thought that this one network represents a microbial ecosystem of 10 years. How can one network represent 10 years? I believe not very well, and this is problematic. We have a static network to represent a dynamic system. My solution is a dynamic network. Using the abundance data as well as association information, I generated subnetworks from the static network. For each month, I generated one network. Here you see that the dynamic network seems to undergo a cycle. It's 10 years, and we observe a pattern and reoccurrence of the same edges. What I find even more fascinating is that now we can study networks in time, and through it, the microbial ecosystem in time. One of the first things I measured was network similarity with the Jaccard index based on a set of edges. If two networks have exactly the same edges, the score is 1. If two networks don't share any edge, the score is 0. I computed it for each pair of networks. We found an overall similarity, which is rather low. And as seen on the left, this didn't change when comparing networks within each year. However, the average similarity increased when we compared networks intra-seasonal, as seen on the right. Our results point to a seasonal dependent recurrence in network architecture. It is ranging from modest to moderate. Next, we used a more sophisticated approach, graphlet correlation distance. Since the start of my project, I was envisioning a network of networks, and I'm very proud to present it to you today. Each node represents one of the subnetworks you just saw in the video. And If two nodes are connected, it is because they are similar according to their graphlet distribution. Here, I colored the nodes according to the seasons as classified in my previous study on the same dataset. I also determined modules, I found 4, and I mapped temperature as it has been shown to be a major factor driving microbial abundances. As you see, and as expected, it aligns well. Lower temperatures in the winter months, and as we go through the network of networks in a clockwise direction, we reach the warmer months in our time series. Next steps will include marine microbial interpretations. Here, I presented the approach of generating a network of networks. There are various applications, so if I sparked your interest, let's have a chat. To conclude, we quantified network reoccurrence in a model marine microbial ecosystem. There was a low amount of interseasonal recurrent edges, but a modest to moderate amount of intraseasonal edges. All in all, our results indicate that ocean ecosystems require at least a moderate amount of recurring microbial interactions to function. I want to acknowledge Ramiro Logares and Anna Scrabero. Thanks for your attention, and let me know how a network of networks could benefit your studies.